Hello, we are Team 7 and we will be presenting assays for cytotoxic T cells. The golden standard for determining cytotoxicity of T cells is the chromium release assay. This type of assay is carried out by labeling target cells, which can be any type of cell, with chromium-51. These labeled target cells are then incubated with effector cells, which are T cells. The effector cells will lyse the target cells, which releases the chromium-51. Chromium-51 is detected by a gamma counter and indicates the cytotoxicity of the T cells toward the target cell. Important parameters to control for this assay are the volume of media or buffer, radioactivity for target cell labeling, and the effector to target cell ratios. The purpose for controlling these parameters is for optimization. The chromium-51 that is released from the target cells after lysis is counted by a gamma counter. This is because chromium-51 is a gamma emitter. This means that the number of lysed target cells can be counted and then compared to the population number of target cells. The data is then analyzed as the percent lysis, which indicates the percentage of target cells that have been lysed. The percent lysis is proportional to the cytotoxicity that the T cells have towards the target cells.
So the main goal for using assays of cytotoxic T cells is to cause and measure the death of a target cell. The most popular T cell is the CD8 T cell, and the most popular use for this technique is for cancer treatment in order to kill and target cancer cells. To the right, we have a depiction of what exactly would happen when the T cells attack the host cell. So first they would bind using surface membranes, which would lead to the production of lyse molecules. These lyse molecules will have the potential to form holes in the membrane of the host cell, which would eventually lead to the host cell's death. In the next 3 slides, we will have an example of how this technique is used in order to kill breast cancer cells. So first, you would have the important components. Obviously, the CD8 would be involved. The HL class 1 molecules would be the components that will contain the surface membrane to attract CD8 to the breast cancer cells. And then we would form the granzyme and preferrin lytic molecules inside the CD8 T cell. First, we would have to prepare the T cell lines, separating them from the blood, and this would take approximately 6 days. We would also have to prepare the breast cancer cells using the Xceligent software. And then finally, we can have both of the cells interact, the tumor cells and the T cells, which would be in this procedure. And then we would have the chromium release assay, which would help us quantify our success and also the percent lyse calculations to make it a concise presentation.
Moving on to the quantitative aspects, activated CD8 T cells destroy any cells that exhibit the unique MHC class 1 complex that they identify. Thus, the simplest and most quick T cell bioassist, which is the killing of target cells by cytotoxic T cells, can be used to measure CD8 T cell activity. As shown in the figure below, chromium-51 release assay is used. Radioactively tagged sodium chromate is taken in by live cells and not released spontaneously. The radioactive chromate is produced when these tagged cells are destroyed and its presence in the supernatant of mixes of the target cells, thus quantifying cytotoxic T cells. To further explain this image you see, target cells are radioactively tagged with sodium chromate, then washed to remove excess radioactivity and then exposed to cytotoxic T cells. With this, after 4 hours of mixing target cells with T cells, cell death is quantified by using the release of radioactive chromium into the media. Intact experimental animals can also be used to measure target cell death by cytotoxic T cells. This obtains to assays done on pathogen-infected mice known to induce a strong cytotoxic T cell response. So an antigenic peptide is incubated with target cells, which binds to MHC class 1 on the cell surface. After that, the cells are targeted with a low dose of fluorescent dye. A control population of cells that have not been administered the antigenic peptide is treated with a high dose of fluorescent dye to differentiate them from the antigenic bearing target cells. The two cell populations are combined in a 1:1 ratio before being injected into the testing animals. Cells are then extracted from the animals 4 hours later and examined using FACS, which is flow cytometry. This allows the specific target cell alias to be computed using the ratio of two fluorescent dye labeled cell populations. All of these quantitative aspects quantify the number of cytotoxic T cells in order to provide an analysis of the effects that are being carried out on the immune system. Thank you all for listening.